Today, 20% of cancer patients benefit from immunotherapy, and we don't have any good tools to predict who would respond and who would not respond to these great therapeutics. To overcome this challenge, we have conducted a study that aimed to identify spatial signatures from biopsy slides that were taken from lung cancer patients at diagnosis in order to predict response to immunotherapy. To do that, we've analyzed biopsies from more than 100 non-small cell lung cancer patients stage 4 that were treated with Keytruda and extracted hundreds of different features related to the interaction between immune cells and tumor cells in order to find the right maps to distinguish between patients that benefit and patients that do not benefit from Keytruda. After generating immunological spatial maps for each patient and correlating them with their response, we were able to build a classifier that identifies responders. Our results showed that patients that had a positive nuclei score had a significantly higher survival and benefit than patients that had a nuclei negative score. While patients with positive score had 70% chances being alive 2 years after starting the treatment versus only 33% at the negative group. After correlating those maps that we have generated from the biopsy slides with the clinical outcome, we have found that certain spatial features were actually highly predictive of immunotherapy. For example, the level of interaction between the T cells and the tumor cells in the tumor microenvironment was highly predictive for response. The impact of such biomarker is enormous. Having such a great tool will allow physicians to better choose the right treatment to their patients and the pharma companies to enroll the right patients to their clinical trials testing the new generation of immunotherapies.